on this computer. And then I'll start the webinar. Let's share my screen. Good afternoon, everyone, or morning, depending on your time zone. We're just gonna give it a minute or two for more people to log in, and then we're gonna kickstart our webinar. Hi, Dominica, can you see the screen fine? Dominica?
Yes. Yeah, I can see.
Okay, I think we can start now. Good afternoon or good morning, everyone. My name is Anzara Mabrana from South Africa. I'll be co-hosting with Miss Dominica Berardi, and our two speakers for this afternoon are Dr. Msizi Mhlongo and Ms. Pfano Maphari, both from the University of Johannesburg, and they will be taking us through metabolomics at the forefront of plant microbe interactions and phytochemistry. Ms. Dominica will introduce Ms. Pfano Maphari later in the session, and I will just start off by introducing Dr. Mhlongo. But first, please check out our social media pages. We have a Facebook page under the Metabolomics Society. And on X, formerly known as Twitter, we have the EMN page that you can please follow us for any EMN related activities. And then we have the Metabolomics Society Twitter page or X page that you can also follow for all the other updates. And then if you have any inquiries or would want to reach out to the Metabolomics Society, you can reach us through the email. And as I have indicated, these are the speakers for our afternoon today. And then I will just hand over to Dr. Mhlongo shortly after I read out his bio. Dr. Msizi Mhlongo is an independent researcher at the University of Johannesburg in South Africa. In 2015, he obtained his master's degree in biochemistry and received the Best Master's Student Award in the Department of Biochemistry. In 2016, he received the S2A3 bronze medal from the Faculty of Science for his contribution to the advancement of science through his master's project. In 2018, Msizi was appointed as an assistant lecturer in the Department of Biochemistry, and in 2020, he completed his PhD in biochemistry. Upon the completion of his PhD, Msizi was appointed as a lecturer in the same department. He was also promoted to a senior role in that particular department. His research activities are divided into two categories. The first one, plant-microbe or microbe-microbe interactions. And then the second one, phytochemistry and discovery of drug-like products. Dr. Mhlongo was also part of the EMN a few years ago, so we are excited to have him back sharing some of his research. Over to you, Dr. Mhlongo. You can start sharing your slides and then take over for the presentation.
Greetings, everyone. In a second, I will just deal with the technical issue that I'm experiencing. Then I will share my screen. I'm not so sure if it's visible on your side.
Yes, it's visible.
Can you all see my screen?
I just went away right now. Please read.
Okay, let me try again. Can you see my screen now?
Yes.
And my presentation?
Yes, you can just go. Perfect.
Okay. Greetings, everyone. Thank you very much for the introduction, and I would like to thank the Metabolomics South Africa, the International Metabolomics Society, together with the EMN, for allowing them the opportunity for me to present the research that I've been doing since the 3 years I started research at UJ. So in short, my name is Msizi Mhlongo, and as been introduced, I'm a senior lecturer in the Department of Biochemistry, and this year I served as a Deputy HOD for teaching and learning in the same department. And today I'm very pleased to be sharing with you my presentation, which I decided to title Plant Growth Promoting Rhizobacteria, the Messiah of Plant Growth Protection and Sustainable Agriculture, or Sustainable Farming. I had to think a bit far or a lot about the topic, and I chose the term Messiah because these organisms seems to be able to do it all when it comes to plant protection, plant growth promotion, and also the potential that they possess in agriculture that you can use them. So please excuse me, I'm a bit nervous, but I will try by all means to remain calm throughout the presentation. This is the overview of my presentation. Just need to get my pointer. This is the overview of my presentation, which I will start by just giving a brief information about the University of Johannesburg, the metabolomics at UJ, the problem that we face in terms of food security versus the population, changes in global food, challenges that we have in global food production. I'll introduce plant microbe interaction, plant growth promoting rhizobacteria, and then I'm going to use a series of studies that we did on wheat priming by PGPR on striped rice to explain why these organisms sort of hold the potential in agricultural applications. So the University of Johannesburg is a public university that is situated in Johannesburg in South Africa. It came to existence on the 1st of January in 2015 as a result of the merge between the Rand Afrikaans University and the Technikon, the Westvleteren Technikon, together with the Soweto and the East Rand campus in the Vista University. This is a vibrant university. It's multicultural and dynamic. It's sharing a space with the Johannesburg, which is the city which houses this university, and it's a proudly South African university, which is well prepared for its role in actualizing higher education's potential in the continent and also in the country. The university houses a total of 8 faculties that are spread throughout 4 campuses, and the Department of Biochemistry is situated in the Auckland Park Kingsway campus, which is the main campus of the university. And when you take a look at the university, you can see this is our campus that we are, and the Department of Biochemistry is found here on the C-Labs on the top floor. So this is where you will find the Department of Biochemistry. Okay, so back to metabolomics at UJ. In 2008, Prof. Duberry, in collaboration with the CSIR, received an NEP grant for the SYNAPT G1 UHPLC MS. This instrument has been with us for almost more than 14 years and has graduated a number of students, of which some are not depicted here. But these are some of the students that remained in research. So the pioneer of metabolomics at UJ was Prof. Duberry since 2008, and then he has trained a number of students, but I would like to highlight 2 that are still involved in research. One of them is Prof. Madala, who has since moved to University of Venda, hence I used the dotted red lines there. The red lines just indicate the students that were trained by Prof. Duberry in metabolomics. Dr. Tukizimana is still with us here at UJ. I put myself a bit lower there, and I've got those green arrows because the 2 have poured their knowledge to me as I was a student. So I'm sort of behind them. I came after them and I joined the metabolomics group. And then I have blue lines there that are dotted lines, which shows that I still have collaborations with the 3 pioneers of metabolomics at UJ. The gray lines, the dotted gray lines indicate collaborations that have been included. Prof. Duberry together with Dr. Tukizimana. They co-supervised one of my master's student who's now doing PhD. And this project is one of the results that I'm going to use to explain the use of PGPR in agriculture. And then the solid gray lines just indicate the ongoing collaboration between the 2. As you can see, Prof. Madara still supervised 2 of my students in phytochemistry and natural product research, and also one of my students also co-supervised by Dr. Tukizimana. So my research, I'm a very ambitious person. So my research spans a lot of areas in plant-microbe interaction, where we try to elucidate the action mechanisms that are involved when microorganisms interact with one another, either the microorganism being a pathogen or being a beneficial microorganism. Also this year, with 2 of my students, which is Nompumelelo together with Gamo, we started to introduce something that is called microbe-microbe interaction. We are trying to elucidate mechanisms of action between these microorganisms, be it 2 PGPR microorganisms that interact with one another, or be it a PGPR together with a pathogen, just to see how these microorganisms interact with one another. Then on the phytochemistry side, I've got a lot of master's students, but here, this research is still at its very, very early stages. Pfano will share what we've been able to accomplish so far, but it's mostly the profiling of the plant medicinal plants that are found in South Africa, trying to elucidate the action mechanisms using computational tools such as network pharmacology and molecular docking. However, we still look for collaborators and everything so that we can really take this further, and maybe it could result to the discovery of other drugs that can be used to treat various diseases. So back to the business. We know that we have to produce and maintain human survival on Earth. However, the rate at which we are producing is alarming. And this rate, when it's starting to be compared with the food demand, every time we produce, a new person is introduced, that person put pressure on the food production, and that sort of threatens the food security that we have. So studies have shown that in order for us to meet the food demand that is depicted in 2050, there should be a rapid increase in food production. One of these, especially the developed countries, Those countries don't need a lot of increase. They just need around about 12%. But countries, especially in Africa, that are developing, which are also some of the countries that are sort of living in poverty, we will need approximately 115% of increase in food production in order to meet that demand. Furthermore, the land, the earth, as we continue to grow, the population and everything, the arable land remains the same. As you add more people, the hectare per person also shrink. As you can see here, that the number of people continue to increase, but when you look at the hectare per person, it's also going down. This means that we'll have less land to produce food. At the same time, we'll have a high demand of food from the population that we have. So there is a necessity for interventions in order to make sure that by the time the population reaches 2.5 billion, we'll be able to reach to produce enough food for all. So the food industry, which is mainly the primary industry, is the agricultural industry that deals with the farming, the production of food, either big crops or animal or anything. They all depend on crops. The reason for that is that plants are able to harness the energy from the light together with carbon dioxide to produce different compounds that are suitable for their growth and development, and also we benefit from that as humans. So it's important that we meet these challenges that plants face on Earth. So in nature, plants are faced with a different of stresses ranging from biotic and biotic stresses that impact their survival and their growth. So these have a negative impact, and when you look at the agricultural industry, this reduces the output that the agricultural industry can have. So, and it is known that plants rely on their innate immune system, but however, this is not always successful. So the plant can either get diseased from a pathogen or succumb to whatever stress that they are experiencing, and that also reduces the output from agricultural industry. Number two is that we switched as humans, which I believe we are the most problematic species on Earth. We always find the simplest solution that we think are the best solution, and few years down the lines, we then discover that these solutions are not the best. So to combat all of this, we either did resistant breeding, which allow us to use extensive use of agrochemicals. Also, we've got plants that are resistant to different environmental conditions. The use of agrochemicals has been on the rise and everything. All of this from genetic modification and the use of agrochemicals has led to environmental concerns, as they are detrimental to the environment. Thus, there is a need for potential sustainable solution. One of these is the good microorganisms that are found in the rhizosphere or even within the plant that have been shown to have a better solution for agricultural expansion. However, the use of these organisms is limited because there's no sound science that support their use, and it's mostly based on people telling each other that this one performs better, this one doesn't perform better. So there is a need for us to investigate the scientific mechanisms and also to support these studies and the use of these organisms, also to help the government to come up with legislations that could force farmers to sort of switch to these organisms. So in order for me to introduce plant growth promoting rhizobacteria, I decided to select 2 review articles that we've written. One was written by me in 2018 when I was doing my research, and the other one was written by my student Dennis recently during his master's study. So if you want to know more about plant growth promoting rhizobacteria, please feel free to go and read these articles, but I'll try by all means to cover as much as I can. Okay, so as we know that in nature, plants encounter different microorganisms. These microorganisms can either be non-pathogenic, they can be pathogenic, and they can be beneficiary microorganisms. The interaction differs depending on the organisms. So we all know that plants have a preformed defense, which is the first line of defense that prevents the entry of the microorganism into the plant. And these preformed defense are there to defend the plant from this pathogen. What happens is that the pathogen, when you look at the non-pathogenic microorganisms, they can either be stopped by the preformed defense system, or they can bypass it, which means that now they are within the plant. But these are not problematic. When the plant recognizes this, it will activate different forms of defense responses. However, this can lead to the plant being tolerant or being induced for protection against other microorganisms. These ones do not have any effect on the plant. So the plant will eventually come back to homeostasis to realize that there are no other damages that are occurring in the plant due to the presence of these organisms. The other ones are pathogenic microorganisms. There are two events that could happen. When the pathogen infects the plant, it can bypass the first line defense, induce a defense response. However, that defense response can be insufficient, which means that it can cause an infection, which then could lead to disease, which means that the plant now is in a disease state, and it will then succumb to the infection from the pathogen. On the other hand, if the plant is stronger enough, it can then survive the infection, and that would leave the plant with a memory, or a memory to sort of also defend better against the same pathogen or other pathogen in the near future. So the plant after that is then said to be tolerant or resistant. The third form of microorganisms that happens is that there is the beneficial microorganisms. These microorganisms can either be found on the surface where they perform their beneficial effect to the plant, or they can also be found within the plant. The interesting about these organisms is that they do not cause any harm to the plant, but they support the plant. There are various chemical events or biochemical events that takes place that allows a successful interaction between the plant and the PGPR. And this is driven by mostly of the metabolome. And once this interaction is in place, the metabolome becomes very complex, because it then becomes difficult to see which metabolites come from which organism. And we are stuck here as scientists because we cannot really tell whether the PGBR, once these organisms interact, which metabolites come from which organism and everything. And that's where we are stuck. But there are tools that have been developed that are able to help us with that. So, as I said, that these organisms facilitate plant growth. As you can see that they are mostly found in the rhizosphere. Those are the ones that I deal with. The plants secrete root exudates. The root exudates recruit different PGPR, which perform various functions. One of them being nitrogen fixation, solubilization of nutrients, and decomposition of biological material to make sure that the nutrients from the soil are also available. And then this results in increased photosynthesis, which means that the plant grows better. It can photosynthesize, it has more energy, can acquire more energy from the sun, and then it can grow. And also the plant has an increased biomass, which means that the plants that have been treated by PGPR have been shown to be bigger compared to plants that are not treated with PGPR. Furthermore, the yield output is also increased. Studies have shown that you get around about 20 to 40% increase in yield, which is good for agricultural industry. Another benefit is the induction of resistance against the environmental stresses. So these organisms, as I said, that they are messiahs, they seem to have a vast number of organisms of benefits to the plant. So how these organisms interact, or what events that takes place when these organisms interact? There are events that takes place in the rhizosphere. The first thing is that the reduced exudates will recruit specific strains, and these strains will be recruited by the root exudates. From there, these organisms can also recruit other microorganisms through chemical secretion. So they secrete other chemicals that can recruit other microbes, and that then builds the population of microorganisms that is found in the rhizosphere. Moreover, these organisms can have inhibitory effect on pathogenic bacteria through the release of also signaling molecule, antibiotics, siderophores, and so forth. And then they can, you have also the soil-borne pathogens, which could be effective affecting the plant and so on. So these organisms can help to prevent infection from soil-borne pathogens. So all this interaction that takes place in the rhizosphere result to metabolic perturbation or biochemical perturbations event, or biochemical events that happens in the crop plant. For priming purposes, there are 3 steps that you discover. One is the signaling. Once the organism is perceived, or the signal from the organism is perceived, there are signaling events that is driven by phytohormones and other signaling molecules. These initiate defense responses and everything. And then these defense responses will result in the modulation of the primary and secondary metabolites. And then this renders the plant a faster and stronger immune response to a broader spectrum of resistance. It could be your pathogen, it could be abiotic stresses or biotic stresses, and then the plant is said to be primed from that. Also, it could drive interaction, what we call allelopathy, to other organisms. So when a plant is treated by one, it can also notify the neighboring plant of what is happening. As I said, that there are cellular events that are taking place between the two interacting organisms that result in the mutual relationship between the two. Some of these mechanisms are not really elucidated. or they are not really known. Thus, in one of the review, we then postulated this diagram. So what happens is that the plant itself will secrete what we call root oxidose through transporter proteins to the rhizosphere. These can be picked up by the microorganism through, there are receptor proteins that could facilitate different signals, or they can diffuse through the membrane and they can drive the cell metabolism of the PGPR. There are events that are really not known at the moment, but these events lead to expression of certain genes that are related to the production of phytohormones, certain volatiles and siderophores, antibiotics, and other molecules that are beneficial to the plant. And these will then be secreted outside through transporter proteins, and they are secreted to the rhizosphere. The plant can pick up these signals that are processed there, and it leads to different cellular events all the way there, and then it can lead to induced resistance. So this is just a simplified diagram that we came with, but there could be other events that are taking place. So that's why science is still needed in order to elucidate the action mechanisms of these organisms. So enough about PGPR. Let's come back to the series research that I'm going to study. In this study, what we did, we wanted to see the priming effect or the ability of this microorganism to induce resistance against a stripe-rust fungal disease on wheat. So why wheat? Wheat is the second most produced crop worldwide, and it's processed to different products that we eat on our daily basis, and also it's a stable food for most of the countries. In recent years, it has been seen that the production of wheat is declining, especially in South Africa, and this is due to climate change, reduced in arable land, and diseases such as stripe rust. So this is just the overview of the global production of wheat in South Africa. As you can see that the area harvested is indicated in red, and you can see that as years goes by, the area becomes smaller. And you can see that even though the yield is increasing and increasing, stripe rust is sort of becoming more and more of a problem in South Africa. And thus, we decided to do this research. So stripe rust is caused by the Puccinia triformis triticii, which is referred to as stripe rust. It's a fungal disease. It thrives mostly in cool and moisture environment. And in susceptible cultivars, it can cause 10 to 100% food losses, yield losses in crop plant. And it was reported in South Africa in 1996, and in a sub-Saharan region, Zimbabwe is the last country to be added into the list of countries with this disease. So far, resistant breeding of cultivars and also plant breeding are the main mechanisms that are used. These either have resistance gene that are resistant to herbicide that are applied or agrochemicals that are applied to combat the disease, or they've got resistance genes that are resistant to the pathogen itself. The pathogen affects the leaves and reduces the ability of the leaves to photosynthesize. As you can see here, you've got the dead leaves where it has killed some of the leaves, and also you've got these spores, orange spores that cover the leaves totally, which means that now the plant will have reduced photosynthesis, and when that happens, it means that the plant cannot grow well, and that then will lead to reducing yield. So this disease is said to be an airborne, which is becoming most of a problem because sometimes in South Africa, we've got wheat that are grown in winter and some that are grown in spring in different seasons. And we know some seasons are windy, some are not. So it can spread to other croplands to wheat. And when the environments are unfavorable, it can also survive on voluntary plants until the environment are very favorable, and then it can start infecting wheat plants again. So fungicides and everything have becoming the main mechanisms of controlling this pathogen, and also the breeding of resistant cultivars. As I said, the use of agrochemicals is in problem to the environment, and there is a need of sustainable agriculture, and the PGPR comes into play. So this was a series of a study that we did. The aim was first to use metabolomics to see if we can be able to distinguish between susceptible cultivars and resistant cultivars. The other one was to investigate the metabolic perturbation occurring on the rhizosphere of plants treated with PGPR, and also on the plant metabolome treated with PGPR. The other one was just to compare two susceptible cultivars from the cultivars that we used to see if we infect these cultivars with the pathogen, what are the metabolic what are the metabolic events that are associated with resistant cultivars and susceptible cultivars. And the last one was to study the priming effect against the stripe rust. So on onset, as I said, we started with the metabolic comparison. Here we had 4 cultivars that we obtained from ARC in Bethlehem. These cultivars has a varying resistance and susceptibility to aluminum toxicity and also stripe rust. So as you can see, the first cultivar, which was aligned, was susceptible to aluminum toxicity together with the stripe rust. There were 2 resistant cultivars towards stripe rust, which was the Kunap and the Senku. So upon metabolic analysis, we applied the data and then we did some PCA analysis to see the data structure in terms of pattern, similarities, differences between the cultivars. On a 2-dimensional PCA, we could not really classify resistance from susceptible, but when we then did a 3-dimensional PCA, we were able to tell the cultivars that are susceptible to stripe rust and aluminum toxicity, which was Matlabas, Harip, and Eland. And then Kurnap and Sengu, even though Sengu was susceptible to toxicity, to aluminum toxicity, it was resistant to stripe rust, and these you can see them here. The difference observed between the two could be due to their difference in resistance or susceptibility to toxicity. Upon the metabolic annotation, we're able to annotate quite a number of metabolites, and these metabolites belong to the classes that you could see here. Flavonoid was the major class that we could identify, which belongs to phenolics, followed by hydroxycinamic acid, and there were other metabolites that we were able to identify. Then we did a closer look at the type of metabolites that we could identify. The first were the flavonoids. The flavonoids were found to be more abundant in Kunap, which is resistant in both aluminum toxicity and also stripe rust resistance. As you can see here, this is the Kunap one. You can see that the flavonoids were a bit higher on that, together with the green one, which is Senku. Not the green one. Yes, this is supposed to be green, which is the same one. They will be higher. However, when we look at the fatty acid, the opposite was true. The susceptible cultivars, which is Harip, Matlabas, and Eland, were higher in lipid-like molecules. And then lastly, the ferric acid, the caffeic hydroxycinamic acid, were also found more abundant in the susceptible cultivars. So what we couldn't pick here was that there were no distinctive metabolites that we could associate between susceptible or resistance. The metabolites were sort of differentially distributed in terms of quantity among all the cultivars. And it's only based on the accumulation, especially the flavonoids, which were mostly associated with the resistant cultivars towards stripe rust. So these metabolites, such as your phenolics and everything, they span many physiological and adaptive processes in plants, such as morphological adaptation and reproduction, development, and defense against abiotic stresses. When it comes to defense, they can protect plants against oxidative stress by scavenging free radicals that are results of reactive oxygen species. They are said to be antimicrobial, which means that they can inhibit pathogen growth. They can also be incorporated into the cell wall so that they strengthen the cell wall to prevent more infection to the plant. And lastly, they can chelate metals, which is one of the mechanisms that you can use in order for crops that are mostly resistant to aluminum. So they are able to chelate these metals in order to reduce their effect. Also, metals can lead to reactive oxygen reactive damage, which also these crops these metabolites can also protect against. The other class that was mostly found in susceptible cultivars was the unsaturated fatty acid. These, they maintain the cell because they are formed as the constituent of the cell membrane, so they help to maintain the cells. They also serve as precursors for the biosynthesis of phytohormones such as jasmonic acid. Jasmonic acid is a phytohormone that is known for plant protection against abiotic and biotic stresses. And also they can be signaling molecules and regulatory molecules. They bind and regulate receptor proteins that control major networks, such as your cellular metabolism and signal transduction that could also be related to defense response interaction. So overall, this study that we have, overall, the outcome of the study revealed that significant metabolite variation in organisms could be both qualitative and quantitative, as many metabolites were common among the 5 cultivars compared. Metabolic differences in the wheat cultivars resulted in apparent cultivar-associated differences based on genetic differences between the cultivars, and genetically related can vary in metabolite contribution, composition, and distribution attributed to the phenotype and the functions of agrochemical properties. In addition, some distinct metabolite markers were identified from the selected wheat cultivars. For example, quinic acid, valine were identified as metabolic markers for the susceptible Senku cultivar, along with the unsaturated fatty acid. And at some point, the QNAP, which was the resistant cultivars, showed a higher accumulation of flavonoids. So these were just the summaries that we had on the study. And then we moved on to the other study, which was now to look at the metabolic differences that are happening in the rhizosphere due to PGPR treatment, and also to the plant, which were treated by 2 PGPR. So I'll try to move fast. I think I'm falling behind time. The study was divided into 2, the rhizosphere metabolome together with the plant metabolome. And then this was then analyzed through mass spec and multivariate data analysis and together with molecular networking. So from PCA point of view, we were able to see that the first one is the harib, which was the susceptible cultivar that was chosen, and the second one is the kunap, which was the resistant cultivar that was chosen. These were then treated by 2 PGPR, which is Bacillus subtilis and Epine Bacilli alveni. And as you can see in A, the untreated were clusters away from the treatment ones. And then we also have some time-dependent clustering that was observed when we look at week 1, week 2 on the different cultivars. And then we did some molecular networking. From the molecular networking, these are the classes of compounds that we could identify, ranging from organic acid, your benzoate, your lipids, and everything. Benzoate was the most significant class of metabolites that was found. It is a metabolite that is sort of a taxonomic metabolite that is mostly found in grass families and plays a different role. This was discovered in the rhizosphere. The phenylpropanol pathways, they also found in the rhizosphere, and they perform different functions from antimicrobial to recruitment of other PGPR and everything. And the lipid-like molecules could play a different role, such as protection of microorganisms in the soil, recruitment of them, and also they serve as a lubricant that allows roots to penetrate the soil without damage and so forth. Benzoate are interesting because they also reported to recruit microorganisms. At the same time, they also reported to inhibit soil-borne microorganisms. The organic acid can be suggested to be just organic molecules that are used by the interacting organisms or by the PGPR or by the plant secreted by them. They are just there to serve as nutrient sources for the interacting organisms, especially the PGPR. So these were some of the studies that we have, and then we can move on to the plant now. We then look at what effect do these PGPR have on the plant, and I did some data acquisition. Similar approach were then seen, where your untreated was found to cluster differently from this is the susceptible cultivar, this is the resistant cultivar. You can see that your untreated, which is blue, clusters away from the Bacillus subtilis, which is in red, and the Bacillus the Epine Bacillus, which is in green. And then we then did some time trend to look at what is happening here. You can see in green is the harib treated with Bacillus subtilis. Here is harib treated with the Epine Bacillus. Also here you can see the untreated there in purple and red, and also the other different in terms of time and everything. So there was a time trend that was observed, which could be postulated to the differences in the metabolome that were observed thereafter. So we did some also molecular networking. These are the classes of molecules that we found. Your lipid-like molecules, which could be incorporated into the cell wall. They also have antimicrobial activity. Your organic acid derivatives, and together with the phenylpropanol pathway. When we then did the comparison between these, you can see that you've got the resistant cultivar in red, and also in green, treated by the 2 bacteria, and then this is the untreated. The resistant cultivar, susceptible cultivar, which is HARIP, showed a higher accumulation after the treatment of these metabolite that are observed here, which belongs to flavonoids, some tyrosine there, which is very, very important for the phenyl propanoid pathway. So these are some of the metabolic changes that were induced by the Bacillus subtilis. On the other hand, the pine bacillus, which is this one here, when treated by that, it causes the accumulation of mostly cinnamic acid derivatives, benzoic acid derivatives, and some of flavonoid related molecules. So what we observed here was that treatment of the susceptible cultivars leads to the production of phenyl propanoid pathway, which is very good, that you will see when we look at the defense responses of these cultivars to the pathogen alone without priming. And then we did some pathway mapping and everything. That there are 5 pathways, 6 pathways that we found. Most of them were the citric acid pathway, which just shows you that the energy production of these is then affected by the treatment of these organisms. As you can see, different metabolites were affected there. The second best pathway was the carboxylic and the dicarboxylic acid, followed by the alanine and also the phenylalanine, tyrosine, and tryptophan biosynthesis pathway, which is presented here. This pathway is very, very important because it drives to the production of the phenyl which are molecules that are known for the defense mechanisms when it comes to plant. Diverse and the complex metabolome of the rhizosphere and the PGR treatment induce metabolic changes in both the rhizosphere and the above chemical space of the plant. These belong to the phenyl propanoid pathway and so forth. And therefore, we postulated that this study could serve as a basis for further studies to elucidate the action mechanisms in the interaction below the rhizosphere. The third study that was used was the metabolic profiling of susceptible and resistant wheat cultivars responding to the pathogen alone. And same time, we did some PCR analysis. You can see differences between treated and untreated of the 2 cultivars, Kunap and what. And then you can then see a time dependent. What we observed here was that the untreated, which were naive plants that were not infected, they had sort of their own time trend, which could be due to growth of the plant in different weeks. As you can see, we sampled from week 1 all the way to week 4. And then we also observed that when you look at week 1, you can see there's a clear difference between the treated and untreated, which also then now comes into play to explain that the treatment with the pathogen induced some metabolic differences when compared to the non-treated plant. And then we wanted to investigate these metabolic differences. We then did some annotation of metabolites and everything. The resistant cultivar here showed a quick and faster accumulation of metabolites that are involved in defense, such as your flavonoids, signaling molecules and everything, whereas these metabolites started accumulating later in the susceptible cultivars. And then this then goes to say that the presence of flavonoid that was observed in the resistant cultivar play a role in the defense response of this one, whereas these metabolites started to accumulate a bit later in the susceptible cultivar. Then the last study that we did was to evaluate the priming effect of these PGPR2 PGPR strains against the pathogen. We did some PCA. We could see some differences between the treatment and everything, even though they were not apparent. But you can see that the naive and control compared to the PGPR treated and everything, there was some sample grouping and everything. As you can see here, you've got the control there, which was week 1. And then you've got week 1, week 2, week 3. And then these cluster differently from the PGPR treatment treated organisms. Plant. It's important that here we only use this as a susceptible cultivar after we observed that when we primed this one, it leads to the accumulation of flavonoids that were not observed to be higher in it when compared when we did the cultivar comparison. Also that we saw that the accumulation of this flavonoid is sort of delayed when you infect this pathogen, this cultivar with the pathogen. So we then went on with the susceptible cultivar add-on here to investigate what is the PGPR able to prime this cultivar against the pathogen. So through morphological results that I didn't show on the other plant, but what we saw here that this is the primed plant. This is the non-primed plant. You can see the spores, the fungus spore on this, and you can see that the leaf is becoming yellow, which indicates that the leaf is about to die. Whereas here, the symptoms that the physiological parameters that were observed here were similar to that of resistant cultivar, which is localized death, and there were no fungal spores that were observed here, which indicate that the PGPR, which was Pioneer bacillus, the T22 induced resistant mechanisms against this pathogen when you compare the two. Also, what we observed was the accumulation of hydroxycinamic acid and some flavonoids that went that high in the susceptible cultivar, which was good because these flavonoids seems to be the ones that play a role in resistance of this cultivar against the striped rust. And then various metabolites were found, and these were found to be differentially distributed, and they span the metabolites that I've talked about. So in summary, this study showed that in the non-treated plant, the organic acid were higher, amino acid went down, and the secondary metabolites, which are sweet specific metabolites, were sort of higher. Were sort of higher, and this is the summary that we had, which says that the TCA intermediate are involved as precursors for synthesis. So you can just read this through. I think I'm over time. With that, there are some ongoing research which various PhD and MSc students are involved in, spanning from plant microbe interaction and all the way to natural products and phytochemistry. I would like to acknowledge my collaborators in plant microbe interaction together with phytochemistry and natural product. Lastly, I would like to thank my student, whom I don't believe this research would have been a success if it wasn't involved them. And here, just as I want to show that they say that if you are a father to daughters, you become soft. As you can see, all the men were not participating in this challenge, but as a father, I was involved there with all the ladies in my group. So I'd also like to thank them. Lastly, but not least, I would like to acknowledge all these companies for their different contribution to my study. And thank you very much. I would like to hand over to Pfano, who will show some information, her research on natural product and phytochemistry.
Thank you very much, Dr. Mhlongo, for your wonderful presentation. I'll hand over to Dominica to introduce Ms. Pfano Maphari, and I would like to encourage everyone to just type the questions in the chat box. Then Dr. Mhlongo and our next speaker will address the questions towards the end of both presentations. Over to you, Dominica.
Yes. Thank you very much for the very brilliant presentation, Dr. Mhlongo. I would like now to introduce Ms. Maphari. She's an MSc student from the University of Johannesburg under the Department of Biochemistry, where research involves the use of LC-MS-based metabolomics in combination with molecular networking to decode the chemistries of asparagus species and further investigate their active compounds and mechanisms of action in alleviating malaria infection. So please join me in welcoming Ms. Maphari, and feel free to start whenever you're ready. Thank you very much.
Thank you. Good afternoon, everyone. Can you all hear me?
Yes, we can.
Yeah, we can hear you. Yeah.
Okay. Good afternoon, everyone. As introduced, my name is Pfano Maphari. And today I'll be presenting to you some work of my research project titled Combining LCMS and Molecular Networking, Metabolite Profiling with Networking Pharmacology to Predict the Anti-Malarial Properties and Molecular Mechanisms of 3 asparagus species in Elevating Malaria Symptoms, which is under the supervision of Dr. Mhlongo and the co-supervision of Prof. Madara, Dr. Kosen, as well as Prof. Simelan. So this is the outline of my presentation. So malaria is a major global health problem causing significant mortality and morbidity annually. It is a life-threatening infectious disease caused by the protozoan parasite of the genus Plasmodium, which is transmitted by the bite of the infected female Anopheles mosquito. There are 5 Plasmodium species that have been confirmed to There are 5 Plasmodium species that have been confirmed to cause malaria in humans, and of these, Plasmodium falciparum is the most virulent and the most prevalent of the Plasmodium species. Treatment options for this disease are scarce and severely challenged by the emergence of resistant parasite, which posed a major obstacle in malaria control. However, modern biologists are now developing selective novel inhibitors for virus disease, that thereby targeting the exploration of natural products. Asparagus is a medicinal plant from the genus Asparagaceae with approximately 300 species that are naturally distributed worldwide. And in this study, we use 3 asparagus species, which are Asparagus dentiflorus, Asparagus facultas, and Plasmosas. These plants are widely used as a nutraceutical, so people use these plants as a source of both nutritional and medicinal purposes. The species within these plants has been used to treat virus alignments such as malaria, pulmonary tuberculosis, kidney infection, eye problems, and infertility problems. And this is because they possess pharmacological properties such as being anti-malaria, antimicrobial, antibacterial, and anti-inflammatory. However, the metabolic choreography of this species is not well understood. Hence, the compounds that contribute to their bioactivities remains unknown. So metabolomics, which is the scientific study of small molecules in a biological system, will be used in this study to explore the chemical composition of these 3 asparagus species. And metabolomics comprises of 4 critical steps, which are illustrated here in figure 4, and I will explain them in more details on the methodology part. Furthermore, metabolomics provides candidate links that can be used as potential drug candidates for drug discovery, which can be through computational tools such as molecular docking and network pharmacology. Network pharmacology is the new scientific method that is being applied for the systematic multi-pharmaceutical study of natural products with desired therapeutic indices. This scientific method can help in understanding the mechanisms of multi-acting inhibitors at many scales, from the molecular to the organism level, by analyzing important features of the biological networks. So figure 5 here is the network pharmacology workflow, which I will also explain in more details on the methodology part of network pharmacology analysis. So the aim of this study was to employ LCMS-based metabolomics in combination with molecular networking to decode the chemistry of 3 asparagus species and investigate their anti-malarial properties and therapeutic mechanism in treating malaria infection, which was achieved by the following objectives. The first one was to profile and compare the metabolome of 3 asparagus species using different solvent extraction, to maximize the annotation of R nodes using the in silico tools housed in GNPS ecosystem, and lastly, to investigate the potential active ingredients and the mechanism of action of asparagus species against malaria using system pharmacology and molecular docking. So for the results and discussion, for the first and the second objective, which is comparative metabolite profiling of different solvent extracts of asparagus species through LCMS-based metabolomics and molecular networking, this is the methodology that was used to achieve those objectives. So basically, the leaves of the 3 asparagus species were blended into fine powders and extracted using 3 different solvents with different polarities. This is because in metabolomics, extraction is considered as the most critical step that has an impact on the recovery and isolation of bioactive compounds in a plant sample. And for efficiency of extraction, several factors such as temperature, pH, time, and the type of solvent used for extractions are considered vital. So in this study, we considered the type of solvent used for extraction as the most critical factor affecting the efficiency of extraction, since it has an impact on the type of metabolites to be obtained. So after extractions, the samples were subjected to an ultra-high performance liquid chromatography quadrupole time of flight mass spectrometry for analysis. The data obtained was then used for statistical analysis, wherein PCAs were generated on Metabolomics. GNPS molecular networking was then used to further visualize the data and for metabolite annotation. And for biological interpretation, pathways and heat maps were also generated on Metabolomics. So figure 6 here is a representative LCMS chromatograms of methanolic, chloroform, and ethyl acetate extracts of Asparagus densiflorus. Here was to show you the distribution patterns of metabolites among the 3 solvents, or to show you how each solvent was extracting. And as you can see, ethyl acetate appeared as the poor solvent for extraction, while methanol and chloroform extracted more metabolites and in a wide range. So to determine the metabolic differences among the 3 species, and when extracted with different solvents, PCAs were generated on Metabolomics. So figure A to C is species per solvent comparison, and D to F is solvent per species comparison. So now looking at figure A to C, which is species per solvent comparison, samples of the 3 species can be seen dispersing in different regions of the scope plot, which indicate that the 3 species are significantly different. And also, when comparing the solvents now, a clear separation of samples can also be observed. However, when you look closely at figure E, the samples of methanol as well as chloroform can be seen clustering at the same point, which suggests that these 2 solvents extracted similar metabolic features as compared to ethyl acetate. So to explore the chemistry of the 3 species when extracted with the 3 solvents, molecular networks were generated. So figure 8 is an enhanced classical molecular networking of asparagus species when extracted with ethyl acetate, when extracted with methanol, and when extracted with chloroform. So observing these results in a topological point of view, it can be seen that the type of solvent used for extraction has an impact on the number of nodes to be obtained in the networks. And also, the type of solvent used for extraction also has an impact on the metabolome chemical classification. And as you can see, ethyl acetate limited some chemical classification. However, it was the one which was able to extract more glycerophospholipid. But on the other hand, methanol was able to extract some of the metabolites which ethyl acetate was unable to retrieve. However, chloroform was able to extract more metabolites as compared to the other solvent. And this results suggest that the type of solvent used for extraction has an impact on the type of metabolites that you would obtain. So to further evaluate the effects of these 3 solvents on the network topology, as well as the chemical classification, the major class of compounds we zoomed in. So figure 9 here is a zoomed in cluster of glycerophospholipids, wherein ethyl acetate was able to extract more glycerophospholipid. And as you can see, there are some of the metabolites which were specific to the solvents, and some of the metabolites which were shared within the species, and some of the metabolites which were unique within the species. Similar approach was observed with the cinnamic acids. However, chloroform was the one which was able to extract more cinnamic acids. Even with the flavonoids, however, here there was a slight variation in terms of the number of nodes. However, chloroform was able to extract more flavonoids, with Asparagus plasmosas being the most dominant species within flavonoids. Similar approach was also observed with the steroids. However, Asparagus densiflorus was mostly dominant in the steroids, and chloroform was the one which was able to extract more steroids. So in the study, a total of 118 compounds. So in the study, a total of 118 compounds were identified, of which the phenolics and the steroids were the mostly dominant classes of compounds. So figure 14 here provides a summary of the distribution of the classes among the three species in which Asparagus plasmosas was mostly dominant in flavonoids, wherein Asparagus falcatus was dominantly in steroids, and Asparagus densiflorus was mostly dominant in steroids and glycerophospholipids. So to determine the variation among the three species, heat maps were generated in which Asparagus densiflorus can be seen mostly abundant in Asparagus densiflorus, and then the flavonoids being mostly dominant in Asparagus plasmosas. So, should I continue? Okay. Since it's time up, I will conclude only on these two objectives. So, in conclusion, this study provided a comprehensive analysis of metabolites in different solvents of Asparagus species, and it further highlighted the molecular networking as the most valuable tool in metabolomics, as it outlined the similarities and differences among the three extracts, even when they were seen to be clustered close to each other through the observation of the PCAs. And moreover, LCMS-based metabolomics and molecular networking, in complement by the right choice of solvent for extraction, could lead to the maximum identification of important metabolites with potential significant to human health, and can be used in future studies for this species. These are my references, my acknowledgements. Thank you.
Thank you very much, Ms. Maphari, for your wonderful presentation. We are going to take a few questions that are in the chat box. If you still have a question for either Ms. Maphari or Dr. Mhlongo, you can just type it up in the question and answer box, and then we'll read them out, and then our speakers will kindly answer them for us. The first question comes from Angigneska Zuba. I know I'm pronouncing it wrong. I'm very sorry for that. The question says you have a lot of, I think this is directed to Dr. Mhlongo. You have a lot of information about the substrates of lignin biosynthesis. Have you analyzed the composition of the cell walls?
Thank you very much for the question. To answer that question simply, we haven't done that, but studies have shown the incorporation, especially the phenyl propanol pathway, that those compounds that are produced there, they can either be directly involved in plant defense, and they can also be incorporated into the cell wall, which then strengthen the cell wall. But in the near future, we might consider analyzing the composition of the cell wall. I don't know if that answers the question. Hello.
Thank you very much for that question, for that answer. The next question.
I can go on. Mm-hmm. Do you want to hand it over, or I can go with the next one if you want?
Yes, please.
The next question that we have in the chat box is, what is the toxicity in terms of percentage of the isolated compound of metabolites? It doesn't specify which kind of compound of metabolite. Can you please specify which metabolite or compound are you talking about in the question that is that it has been asked?
I'll assume this reflects to all the metabolites that Pfano presented on, so Pfano can respond to that. And what is the toxicity of the isolated compound or metabolites? But basically, there were no toxicity studies that were done in this, only in silico tools, which we tried to use them to elucidate the action mechanism of this. But as I said, that this research is still in its early stages. The PhD students that are coming next year will start to incorporate various biological activities to these compounds to see their toxicity, their action mechanisms, and so forth.
All right, thank you very much for that answer. The next question comes from Chinedu. It reads, I think this is directed to Ms. Maphari. Why do you think methanol and chloroform would have similar chemistries compared to ethyl acetate?
Try, then I will support you.
Okay, so that question I'm not quite sure, but I feel like with methanol, methanol can extract a quite range of metabolites from both polar to non-polar compounds, and even with their properties, comparing methanol and ethyl acetate, they are not the same. So I feel like it is one of the reasons which resulted in methanol extracting like quite more metabolites as compared to ethyl acetate. As to why chloroform and methanol extracted similar chemistries, I'm quite not sure.
I will add on to that is that methanol is a polar solvent, chloroform is a polar solvent, which results from the dipole moment introduced by the electronegativity between the chlorine molecule together with the chloroform. On methanol, you've got the oxygen, which also introduce some dipole moment. So those are sort of polar molecules, metabolites, polar solvents. So you would expect that they are not, if you look at the polarity index, they are not far from each other. So hence you could see that they sort of extracted similar or more related metabolites compared to ethyl acetate, which is a non-polar solvent.
Okay, there's another question for Dr. Mhlongo, which is, since you have taken 5 varieties for wheat susceptibility, which variety would be better for use?
Okay. Currently, farmers prefer anything that you say it's resistant to this. So according to the chart that I showed, there were 2 cultivars, but the other one was susceptible to aluminum toxicity. The variety that you could take, considering its performance that we observed, was the QNAP one, which was resistant in both aluminum toxicity together with the stripe rust. But we wanted to see the effect of our PGPR on the susceptible organ. Can it induce resistance that can be comparable to that one of resistance, in order to encourage people to say, instead of you using a resistant cultivar that might require the use of herbicides or other agrochemicals to sort of support its survival, we've got an organism or an organism that you can use in your crop plant that can help to induce some resistance, and that will be that could also induce resistance to your crop. We currently have a field trial where now we are trying to take what we've observed in the greenhouses and trying to translate it to field trial. So we'll then have results to see how these do differ exactly.
All right, okay. Thank you for that answer. The next question is, what type of MSMS system was used to generate the data? and how did you annotate the compounds? I'm not sure if it's directed to Dr. Mhlongo or Ms. Maphari, but either of you can answer the question.
No.
Sure.
This one, can you please update us more on the LCMS method, the software, the type of the instrument, and the data?
Sorry, Dr. Mhlongo. Before that, there are questions on top. The one that I'm asking is from Sebastian Jupiter, which asks what type of MSMS system was used to generate the data and how did you annotate the compounds?
This one. Okay, the type of MS, I'm not so sure what MS, but we've got two systems, which is the UHPLC MS QTOF mass spec that is able to perform MS fragmentation. For Pfano, it was a data-dependent approach, and for my approach, it was the energy ramping with the mass range. So the energy was ramped from a lower energy to a higher energy, and that allows us to generate a lot of fragments. And for the comparison or the identification, MS identification was used where the fragmentation patterns were compared with either databases or some of the compounds that we've annotated in our previous studies. So previous literature was used and everything also to help. Molecular networking, which was able to group these compounds into different classes, was also used, and the output, the data output that we got there was then confirmed using MS fragmentations. I don't know if that answers the question.
Yeah. Thank you very much. The next thing was just a comment from Chinedu referring to his previous question. He just commented that ethyl acetate is a polar aprotic, but chloroform is not. Methanol is a polar protic. EA is more polar than chloroform. I think it was in addition to your response about the solvents, and then we'll move on to the next question. Dominica?
Yes, so I think the next questions have already been answered, but I actually have one question. I was wondering whether in your study, and this is to Dr. Mhlongo, if you can actually distinguish between the metabolites that come from the bacteria and the ones that are actually produced by the plants.
To answer your question, if the metabolite is not specific, like it's a plant-derived metabolite and we know it's only found in plant, you can be able to distinguish between the two. But you find metabolites like organic acids, which could also be produced by the bacteria or be produced by the plant. Those ones are difficult to tell. There are different systems or approaches that people are using. Some of them are radiolabeling, where people are labeling either nitrogen or carbon dioxide. The plants are grown in a closed container with labeled carbon dioxide, and then they start to monitor that labeled carbon dioxide, whether it ends up on the bacteria or anything. Then from there you can deduce to say this one is really derived from the plant or not. But from the analysis that we did, unless you've got pre-knowledge to say this one is from the plant specifically, this one is from the microorganism specifically, you really cannot tell.
Okay, thank you. And also, like another question, you have nicely shown the difference in time of the different treatment. And I was wondering, well, considering, for example, that we want to apply the same treatment to another kind of soil with another composition. And so, for example, another type of pH, how effective that same treatment would be? Do you have any I don't know, like experience or any experiments that you have done by testing different kinds of soils and so like pH conditions?
Not really, we haven't, but studies have shown that if you change conditions, the behavior of the interacting organisms can also change. So for example, you might not get the same results that you get when you grow these plants in a greenhouse versus in field trials. So any slight change in parameters, whether it's pH, temperature, or anything, might result in different results observed.
Okay, thank you. Thank you very much.
Okay, thank you very much.
I think that was it for the questions, and thank you for your great presentations, Dr. Mhlongo and Ms. Maphari. And also to everyone else, we'll be posting about our next webinar soon on the EMN's MedSocs Twitter page or X page. Please be on the lookout for that, and we'll see you next time. Thank you very much for joining this webinar, and goodbye.
Thank you, everyone.
Thank you, everyone. Bye.
Bye.